So this is an exercise to see how our primers would work before we even order them. So let's imagine that we have GapDH primers which we have either designed or we got from publications. So this is GapDH forward and GapDH reverse, and we want to check how they would do with genomic DNA and mRNA transcripts. So to do that, we'll go to PubMed and we'll write GapDH, and we'll go to nucleotides and search for it. And if you will see, it gives us data as genomic. So one sequence is genomic sequence, and there are 4 transcripts. So that means that there are 4 variants of mRNA sequence, and there are 4 variants of proteins. Now let's click on the genomic DNA, and it shows Homo sapiens glyceraldehyde 3-phosphate dehydrogenase, which is on chromosome 12. So this entire sequence will consist of all exons and introns and everything together, right? So we can also analyze this sequence by clicking on highlight features, and we can go down and see that this is coding sequence, and we can change it to exons. So it has 9 exons, and we can click forward button to see all the exons and where they are located, right? So this sequence essentially is with everything, exons and introns, and let's download this sequence in the GenBank format. So we'll just click on send, and from the drop down menu we'll select file, and we'll save it in GenBank format, right? So GenBank format, don't forget that. And now we have the sequence. So we'll click on that, and this will open up in a program which is called Serial Cloner. It's a free program which you can also download, and so now it is imported, and this is the entire sequence. This is how it looks like. What we want to, so let's first save it. We'll say export, and export as GenBank file. So we'll rename this as, instead of sequence, we can put any name that you like. Let's say GapDH genomic sequence, and this .gb will save it in the GenBank format. So now I've just saved it on my desktop, and I can safely close this now. I'll now go back and select, instead of the genomic DNA, I'll select transcripts, right? So it basically, from this genomic sequence you can get 4 different kinds of mRNA, and that's why you get 4 different kinds of proteins. So I'll click on transcript, and this will give me all 4 kinds, all 4 variants of transcript. So I'll just click on transcript variant 1 mRNA. It will open again, and this time, it will only be the mRNA sequence. So I'll save it, and I'll name it as let's say GapDH cDNA or mRNA. Now our aim is to do PCR on these sequences, the genomic sequence and mRNA, to see whether the amplification happens or not, right? So from the top bar of so I'll just remove all these things. So I'll again go to so I'll open this GapDH genomic sequence, and from the top bar, I mean, if you want, you can also see how it looks like. So in the serial cloner, this is how the entire sequence looks like. And this is how the graphic, the map looks like. Now I want to do a PCR, so I'll go to function, and from the drop down menu I'll select run a PCR, and now I'll pick up my sequences. It is GapDH forward, and GapDH reverse. And so we need to also select which will be our template DNA. So from the drop down menu we'll select GapDH genomic sequence. This is our template DNA, and now we'll run the PCR. So the fact that it did give us an amplicon, that means we should have a successful PCR in the lab. And now we'll compare where exactly our PCR primers align. So this is the entire product or entire amplicon, which will be 556 base pair long. So this is our forward primer, and we can actually see that it is ACC, ACA, GDC. Now if you see the reverse primer, you won't be able to find this sequence. This sequence doesn't match with the reverse primer that we have designed, and the reason is primers are always written in 5' to 3' orientation, and the sequence what we have or the reverse primer, we need to find the reverse complement, right? So it's a very simple thing to do. Online there are a lot of programs which will give you reverse complement of any DNA sequence. I'll just paste my reverse primer here and submit, and it will give me the reverse complement, right? So this I will compare with my PCR amplicon, and you will see now it really matches. It's going to be TAC, ACC, AAC, A, yeah. So we can also save this PCR amplicon as, let's say, PCR GapDH. So whenever we actually do our PCR, we know how big the fragment we would be expecting. For example, here the fragment size was 556 base pairs. So this is what we should be expecting. So we can save all these products in our file before we even order the primer. We can do the entire experiment, see how the what the amplicon would look like, and then we order the primers, right? And similarly, we can also do PCR for the transcript, right? So if you want to do real-time PCR, we won't be dealing with the genomic DNA. We will have mRNA product, which will convert to cDNA, and then if you want to see whether primers can amplify that cDNA, so in that case we'll upload sequence 1, which was nothing but transcript variant 1 mRNA, right? And we'll keep the primer pairs as same, and we'll run the PCR. Also do a restriction digestion mapping to confirm that we are getting the right amplicon if we want to. So like here, we can also save this PCR amplicon if we want to. And let's see how to do restriction digestion mapping to confirm that the amplicon what we got is really the one which we are looking for and not something random, especially in the case of amplification from genomic DNA. So to do that, all we need to do is we can from the top panel. I mean, if you click, you can pick out any of the amplicon, let's say PCR of GAPDH from the genomic sequence, and you can click on the graphic map, and here are all the unique restriction sites. You can also select for a particular one to look for if you have REs with you, let's say BAMH1, but unfortunately there is no BAMH1 site here, so you can select one of the So this is a virtual cutter where you can actually cut the DNA sequence and see how the gel pattern would look like. For example, HAE1 is one of the restriction enzymes that we can look for. So we can select the sequence, which is PCR of GAPDH genomic sequence, and then we can cut it with HAE1 and see what happens. So this is HAE1, and immediately it gives a restriction pattern. One of the band is going to be 389 base pairs and other as 167 base pairs, which you can see here. And this is how it will look like against a DNA ladder. Of course, you can always change the DNA ladder. So this way we can do the entire PCR and restriction digestion before we and we can check whether the primers are of the right sequence before we order them. Thank you.